one of the areas that we've done very well in terms of silver linings from COVID has been issues around diagnostics, as well as using genetic data in particular to develop new vaccines and new approaches for prevention. But one of the areas where we perhaps haven't done so well is the issue of therapeutics. So I'm going to invite Professor Lita Chu to the stage. She is the head of the Pharmacy Center at the National Cancer Center here in Singapore, and she's also the chief pharmacist with the Ministry of Health in Singapore. And this session is really going to be focusing on innovations and the use of artificial intelligence and so forth to really do much more precise, individualized trials and so forth. And I'll ask Lita to join me and introduce our next speaker and moderate the session. So over to you, Lita.
Thank you. Very good morning to all of you here, virtually as well. Yeah, very good morning. I think we're going to continue the discussion on drug discovery and development, but from a very, very different perspective on how do we harness artificial intelligence as a tool in this aspect. So, I work in hospital, right? And I can see that whether it's machine learning, whether it's natural language processing or robotic process automation, it's really making its AI as a tool is making headway in medicine as well in the way we deliver care. Yeah. So I thought that it's great that we have our next speaker here with us today, and it's an honor, really an honor to introduce my colleague from the National University of Singapore, Professor Dean Ho. Yeah. Professor Ho is currently Provost Chair Professor, Director of the N1 Institute for Health, and Director of the Institute for Digital Medicine and Head of Department of Biomedical Engineering at the National University of Singapore. Prof. Ho and his collaborators have developed Identif.ai, which he's going to talk about it later, yeah, for the rapid optimization of combination regimes towards SARS-CoV-2 and for pandemic readiness and antimicrobial resistance application. He's really well accomplished. You can see from his CV. And I think most importantly to note that, Prof. Ho is also a subgroup lead in World Health Organization's Working Group for AI Regulation. So happy to have you on the stage, Dean. The stage is yours. Thank you.
Good morning. It's a privilege to be here today. I wanted to thank the organizers for having me. And I think it's also a privilege to follow on from the previous talk, which mentioned the concept and the importance of developing combination regimens to address things like pandemic preparedness and AMR. And so I'm going to start things off today, actually, by introducing the institutes that I direct, and in the concept of multidisciplinary or interdisciplinary engagement, address how we do what we do across a number of different disciplinary domains to address things like pandemic preparedness. And so when I took on these institutes, the first thing we did was to define what impact means for us. I think especially in the context of AI, of engineering, the word impact is used a lot, right? And we wanted to help ourselves understand what do we want to achieve in these domains. And so for us, we wanted to achieve T2 status. And so what do these T levels mean? These are levels of translation defined by the National Academy of Medicine, with T0 representing cells, tissues, animals, all the way to T1 human, T2 patient, then reaching towards large-scale trials and eventually practice-changing healthcare. And our goal was to demonstrate that the work that we do in-house could achieve T2 relevance, all right? And so I think when we look at the Institute for Digital Medicine, we look at N1, I think we've done that. I think we've launched a number of trials that span digital health, digital medicine, to interventional oncology, to infectious diseases and beyond. Due to our interdisciplinary nature, we've been able to not only launch these prospective first-in-kind trials, but to do so not purely from a technology perspective, but from implementation, from accessibility, from economics, behavioral sciences, and beyond. And so before we get to the technology and the AI of pandemic preparedness and antimicrobial resistance, how do we get there? How do we launch new technologies and impact human health? And so this is a remarkable team, and this is only a snapshot of who works with us. But I'm going to highlight a few, right? And so one of them is Johan Sapinel, top left, the first hire to our institute. He's not an academic. He doesn't develop AI models, but he's actually from insurance. And when I brought him in, I told him, I want to launch trials that will impact human health, but we want to make sure we can do what we do at scale. We want to think about accessibility. We want to think about equity. We want to think about achieving adoption, right? So we brought him in, and we've made important strides thanks to his leadership. Dr. Agata Blasiak, a gifted researcher. We also have a team of behavioral scientists because a core ethos of our institutes is that technology alone cannot transform healthcare. And before we think about developing new technologies, can what we develop reach as many people as possible? Can we try to understand the communities that we hope to help? And so we have this team of behavioral scientists, of healthcare economists, implementation scientists, and beyond, to make sure that we don't solely develop technologies without a chance really to reach scale. Right, so today, I'm going to talk about the use of AI for healthcare in the context of pandemic preparedness, AMR, and beyond. And I'm going to start things off with just a few examples of how our technology works, some clinical examples of implementation, and then we're going to talk about pandemic preparedness, AMR, and some other topic areas that I think are relevant. So I want to start things off by talking about AI. All right, I think AI is defined very differently depending on who you speak to. But I think when we think about AI right now, one of the prevalent definitions is that you take a lot of big data. You take a lot of data, you train an algorithm, and you make a prediction, and you hope that that prediction is better than standard care. There's a lot of hopes. You hope that a clinician believes it, the prediction. You hope that the data represents the population you want to treat. And you hope that the data represents equity. And so I'm going to start things off by talking about the fact that we actually totally do not use AI in that context. I'm not going to be talking about any big data-driven AI today. All right, big data AI is important. It's made important contributions to healthcare already, and it will continue to make contributions. The AI I'm going to talk about today actually has no pre-existing data. So when we think about how we acquire that data prospectively to drive what we do, it's a different approach. We're going to be using small data today, data that does not exist when we begin to do what we do. There are a lot of other hopes for AI, including, especially in therapeutics, to deliver the right drug at the right dose at the right time. That's a popular catchphrase for many different fields beyond AI. But it's quite challenging because when we talk about delivering the right drug, how do we find the right drug? What does that mean actually? Is it purely based on mechanism of action? What if you don't have the right drug because of supply chain issues? And also the right dose. What does the right dose mean? Does it always mean higher is better? In the context of combinations, not only can lower doses work better, the doses that you need may change over time depending on who you're treating. Also, what is the right time? There are a lot of variables here. And simply asking pre-existing sets of big data plugged into a predictive algorithm to tell you what to do at a fixed point in time may not be sufficient, right? So we're going to look at something a little bit different. So how do we address this challenge? What you're looking at here is what we call a digital avatar. It looks purely computational, but this is a curve. This is an actual patient driven with real data, driven with prospectively acquired data and very small amounts of data. In fact, we can guide and optimize patient treatment with as few as 3 data points, all right? And I'll tell you later how we do what we do. And with what I'm going to talk about today, we do 2 things, 2 core things. We design optimal regimens either for individuals or for populations, and optimal regimens are defined by actually finding the best possible drugs to put together at the right respective dose. That's what defines an optimal regimen. After that, we figure out how to optimally dose that regimen for populations or for individuals. So this is a prospectively driven, right, not using pre-existing data to drive true optimization. So I'm going to fly through this slide of some clinical examples of how we've done this. These are mostly oncology. We've done ID as well. But I'm going to start things off with the top left. Okay, this is a patient with lymphoma. Wonderful work driven by my research director in the Institute, Ed Chow. This patient had failed about 6 lines of treatment, right, and was about a week or 2 away from a hospice care. Failed immunotherapy, some of the most expensive therapies around. By taking a small bit of the patient sample, and we have no data on this patient to work off of, and using a pool of combinations, right, a pool of drugs that actually represents hundreds of thousands of combinations within 5 days, a completely optimal combination based on the right drugs at the right respective dose was found for this patient, who ultimately achieved a complete response and remission and made it to transplant. Not only did we get a good outcome, this AI platform, again, run prospectively, was able to compare against all other failed regimens and using only this patient's data. We did not rely on anybody else's data to drive this care using this digital avatar approach. We've done this for transplant patients. We've done this for solid cancer patients on the lower left. We recently launched and then reported a trial for gastric cancer patients. And some of these patients who did not respond at all to care, which is quite common, just by changing the dose, without changing the drugs, we could flip patients prospectively from non-responders to responders. But let's talk a little bit about infrastructure, about accessibility. We do not use supercomputing for any of this. We don't use highly sophisticated databases. We use a laptop. We use a laptop computer, small amount of prospectively calibrated data to drive optimal regimen design as well as dosing optimization. Now, for some of them, we need a sample. For dosing, we use small amounts of data. Final one is this lower right patient, stage 4 cancer patient, responding to treatment, but highly toxic treatment. By dropping the dose 50% for this patient, but without changing the drugs, we increase the efficacy, right? Imagine going to a team of top oncologists, asking them to drop the dose in order to increase efficacy. You can imagine what they said at the time, right? But through the course of the last 10 years, right, harnessing this approach, we've been able to make big strides. Small data, prospectively driven. So how do we do what we do in the context of pandemic preparedness and antimicrobial resistance? Right, so I'm going to use the word optimums a lot, right? But I'm going to define what optimum means to us, okay? So I'm going to go back a little bit. When we talk about AI, we often think, we get asked a lot of questions. Can I send you a bunch of data? Can I send you millions of data points, and can you predict a new drug structure for me? Can you optimize a new drug? Can you predict a new target? Can you find a new target? We don't do any of that. So I apologize to anybody who want to come here today to learn about that. We actually don't do any of that work. There's a lot of amazing work being done in that space in laboratories around the world, by companies, but that's not what we do. As I mentioned just a second ago, we've had patients that do not respond to therapies that are otherwise good for them. And just by changing the dose and often decreasing the dose, we flip non-responders to responders. So no matter how good a drug is, when you start to combine them together, it is essential to steward promising drug candidates forward by combining them with the right other drugs and giving them together at the right respective dose. And when that is our aim, we start to reach fairly challenging parameter spaces. We could be talking about tens of thousands, hundreds of thousands, millions, billions, or even trillions of possible combinations. And so what we do is this latter half. We ensure that we steward promising drug candidates forward in an optimal manner. And when we say optimal, we mean we are looking for the best possible drug combinations out of billions or even trillions of possible permutations. So in about late 2019, we put a team together of epidemiologists, clinicians, engineers, healthcare economists, operations experts to develop a platform called Identify, a rapid platform that uses real experiments on live virus, bacteria, etc., to interrogate spaces as large as trillions of possible combinations big, but within days or even a couple weeks to truly optimize best combinations possible and also to identify worst combinations possible, so that we offer this landscape to both prioritize and deprioritize therapeutics. At the time in 2019, we thought, hey, this platform could be useful. We're doing only oncology, but if we can find these answers within days or even weeks, this could be useful for pandemics, which is one of the conclusions we had looked at the time. Right, so what is Identify? What we are doing, because we don't have any data, if I give a pool of 15 possible drugs, which are designed by the clinicians, and I don't have any data, I actually need to prospectively run some of these permutations on live virus and have the live virus tell me which combinations are good, and then further allow the AI to project other combinations that could be good or not good. So we are effectively crowdsourcing live virus to tell us answers from prohibitively large parameter spaces. Again, we don't only come up with a nice list of combinations and then go to clinicians and say, hey, trust us, these could be good. We also answer questions about which combinations could be bad, which combinations or single drugs may be less effective, but not in the context of a small set of experiments, in the context of a trillion possible combinations, so that we put our best foot forward after we run these experiments to look at best and worst possible. Again, we do not use Identify to predict drug structure. It is not a discovery platform. Questions we often get: do doctors believe what Identify tells you? If you predict a combination's good, right, what do they say? And I think one of the first things I'll mention is that, also contrary to traditional AI-based approaches, we don't use a black box optimization where we don't really know where the answers come from, go to a doctor and say, just trust us, the AI is good. The doctors are actually involved at step one. Step one. And what do we do at step one? We ask them, hey, look, something's brewing. Can you come up with consensus, a list of 15 or 20 drugs that are available, that are accessible, that you would actually use in patients if we start developing combinations? So they are actually involved with developing this pool. We ensure that we find the best possible combos out of that pool, right? So it's actually built from initial consensus. And after we do the optimization, we continually consult the clinical team to both optimize and validate. So this workflow here involves prospective, experimentally driven crowdsourcing of live virus to help us build these lists within a few days all the way just to a few weeks. And from this, we come up with a prioritized list. But I want to illustrate this before I show the data, just how big this problem can be. Oftentimes, I get approached by clinicians, and they say, hey, I've got a ton of data for you. Can you predict for me if a 2-drug, 3-drug, or 4-drug combination is going to be good for patients? So they'll give me millions of data points. Unfortunately, that data is based off of fixed dose. And when you look at these millions of data points, it's fixed dose of the same drugs. And so it's not actually, to me, millions of data points. It's only one data point. Because if we look at a galaxy of possible permutations, for example, filling up this room of different combinations, and the optimum is way back there, but all of these millions of data points only represent here, I can't do actual optimization. And in fact, to be frank, you don't even need AI to do this, right, to find this answer. So maybe 3 drugs will be good out of this limited range of possibilities. Let's make things more interesting. Let's look at 12 drugs. Unfortunately, 12 drugs, and I told you dose matters, studied at 10 doses each, that's 1 trillion possible combinations. We don't have the time, we don't have the resources, we don't have the sample to run 1 trillion possible combinations. Make things more interesting. Let's say drug number 5 doesn't work on its own through an initial screen. In conventional drug development, we never think about drug 5 again. We actually keep drug 5 in because you never know. If drug 5 interacts with other drugs favorably, it could in fact represent a vital combination. But how do we get there? Instead of millions of combinations or millions of datas, with about 150 to 200 data points, which we will acquire prospectively from live virus, we will be able to populate the galaxy of possibilities. And from these experiments, we will be able to run an optimization algorithm, which I'll talk about in a little bit, same as we've used for patients before. And with small amounts of data, we will be able to use real data, prospective data, to illuminate the galaxy of possible highly ranked and lowly ranked combinations. But I'll be candid. That list is populated with real data from the dish, but some of the combinations are predicted. After we consult the clinicians about which combinations are of interest, we will revalidate this again to ensure the robustness of the actual data-driven predictions, all right? And oftentimes, believe it or not, we will find that drugs that don't function amazingly well on their own actually comprise some of the highest-ranking data-validated combinations, all right? And so how do we do the optimization? I've shown you a lot of curves. They're nice and smooth, right? And so on the left, back in the day, we ran neural nets on cells. You can kill as many cells as you want, right? And so we ran neural nets on cells, and through hundreds upon hundreds of experiments, we saw that cells can innately correlate drugs and doses as their input, efficacy and safety as their output. Quite simply, inputs and outputs, nice and smooth. We thought we got lucky, right? We thought the neural net ran it on an interesting disease model. We got lucky, so we ran it on another disease model. ID, oncology, metabolic, 30, 40, 50 plus different diseases. In vitro, in vivo, and in human, the surfaces are all smooth. So what does that mean? Why is that important? That's important because if the surfaces are smooth, the AI has helped us discover a correlation that then dramatically reduces the amount of data we need to drive and find these true optimums. And then, quite frankly, the AI evolved itself out of the equation. The AI led us to that relationship, and clinically speaking, we don't actually need the AI anymore to do what we do. Hence, it's AI-based. And so running these small data points allows us to solve these optimums, and these optimums are these combinations defined by best possible drugs put together at the best possible corresponding doses, real data-driven. And from this, we can start to prioritize for our clinical community which combinations they should maybe think about, maybe ones they don't need to think about as much anymore. So what have we done with this platform? First thing I'll note is that when we start looking at these pool of therapies, we get a lot of questions about dosing and concentration. I'll indicate that when we study the concentrations, we do not use traditional EC50 high concentration amounts because that can skew the optimization, right? If you give large amounts of drug to these cells, it can give the false sense that some of these drugs are effective, when in reality these concentrations are so high no human will actually be able to see that type of dose. So we take an honest approach at how we look at some of these drugs. And quite frankly, a lot of the drugs that we use on dish, they're not effective at all as single drugs. That way, when we interrogate this galaxy of possible combinations and these interactions that we need are observed, they are honestly achievable in a clinical setting, right? It prevents over-representation of drug efficacy. And so we've done this to date for wild type, beta, delta, Omicron. We're moving on to the new variants. And what we've been able to see is this dynamic change in different combinations that work for these different strains and also working across strains. And one thing we'll note, as I'll show you, is that our combinations, we go back and see if we can align our results with clinical outcomes without requiring any data from those clinical trials, right? And that's something that we do see. So we worked with an amazing team here in Singapore to drive this. And the next things we have moved towards is knowing that we have this optimization approach, and it's based on input and output. We don't have to build new AI models to go after different bugs. With zero notice, we can apply this to NTM, we can apply this to Klebsiella, we can apply this to tuberculosis. We can immediately apply this as long as there is an assay that the clinicians will believe. And so recently we published more work on NTM, and we've done a lot of exciting work in the space of Klebsiella, and we're moving on to other indications as well. There's something I'm going to mention about, these pathogens, right? There's not only one type. There are different strains, et cetera, et cetera. Something that we do is that we will independently run identify across strains. We'll do discrete optimization across each one, and then we can start to comb this small database here to see if there are combinations that exist that are highly ranked across the different strains. So this gives us a chance to find regimens that are potentially broadly deployable. Another aspect of this approach is accessibility. For a lot of this work that we did, especially for COVID, we spent about 2 weeks per variant to optimize. But we've spent months studying supply chain to make sure that the databases we release, clinicians are able to access and say, hey, if I need the best 2-drug combination that does not contain this particular drug because we don't have it, this database is instantaneously able to provide that answer, right? So that we can iteratively query this database to provide actionable answers because we have combed spaces as large as 1 trillion drug combinations. But again, why believe this, right? I'm going to be very candid. This starts out as in vitro. Why should we believe an in vitro platform to derive quote-unquote optimal combinations? So we have seen results already from our team and with collaborators, and some which I've shown, of taking various indications, starting in vitro, going all the way through to clinical, and those in vitro highly ranked combinations, they stay true. And that's because we're finding number 1 out of 1 trillion, not 1 out of 10. Or 1 out of a small sample set and defining that as the optimum. It's not purely computational. As I mentioned, we don't have starting data. We prospectively drive the optimization process from these huge parameter spaces, right? We can immediately adapt this approach. We don't look at only one regimen iteratively at a time. We look at all of them. Right? And in the cases of urgent repurposing, why only focus on a few regimens at a time when we can comb that galaxy quickly and involve the clinical and the drug development community earlier on at the same time? And so we have an ongoing database. We have an ongoing database of how our results align with clinical outcomes without requiring data from these trials. And some of these are available in some of our papers. We're going to slowly start releasing all of this online. And what's next? Right? We've had a number of countries reach out for the Identify database and achieve clearance for multiple trials using Identify because they have also independently validated Identify results in their own laboratories. Right? And so we've started partnering with a number of entities to do this, both public and private. By the way, for those interested, we've gone on to education as well, right, to talk about the experience of building and validating Identify. For those interested, there's a link over there for free download for a comic book, by the way. Aiden and Eileen versus Vinny the Virus, right, which incorporates a lot of the lessons learned of developing an AI platform to address infectious diseases. Right, free comic book, hope everybody enjoys it. As I move towards the end of the presentation, all right, I know that we're here to talk about therapeutics, But I think as we've seen with the pandemic, there are ongoing challenges that exist. Ongoing challenges like long COVID. And aside from specific symptoms and indications, there are challenges with even being able to run clinical trials, right, due to pandemics. And so, since I run a digital medicine institute, I thought I'd go over 2 slides to talk about digital therapy, which may also find relevance in looking at some of those who have cognitive decline associated with some of the long COVID symptoms, right? And so this is effectively a video game. Instead of dosing pills, we're dosing a game, right, for multitasking. And we've been able to use this game to personalize and decentralize intervention using digital, right? Both for actual cognitive training, but also to decentralize the ability to run trials. My team launched a number of trials during the pandemic, and that's something we've achieved, right? So to conclude, I think what I hope to indicate today is that AI, while a lot of impactful work has been done using big data to drive prediction and drive outcomes, today I wanted to talk about small data AI, the ability to prospectively acquire the data we need to drive optimization. All right, and so final data slide I'm going to show you. Why am I talking about plants now? All right, so I showed earlier that we can optimize combos to suppress cancer, optimize combos to suppress infection. But we know we can optimize combos to go the other direction. All right, and so in the context of planetary health, human health, we know that nutrition content is declining in our food. So we actually took our platform and we started optimizing soil composition to effectively increase the yield of things like spinach by 30%, other plants by 100+%, and independently validate that the nutritional content is unchanged. And so to summarize, much like our medical outcomes, use less ingredients, but get more yield with unchanged nutritional content. So this is a platform we are now expanding to many areas. All right, so I wanted to conclude by thanking everybody for being here, by thanking our organizers, and finally thanking all the audience for listening today. It's a wonderful meeting. Hope everybody enjoys the rest of the conference. Thank you.